Hello guys, today we are here to discuss about the mechanism of urine formation in human being. But before discussing about the mechanism, let's know a slight basic structure of the nephron. So nephron has three major parts. This part this part called as Malpighian corpuscle or renal corpuscle, it further contains two parts, the network of capillaries known as glomerular capillaries, the cup-shaped structure known as Bowman's capsule, and which is followed by a long tubular structure known as renal tubule. This renal tubule, it has further three parts. This part known as proximal convoluted tubule, this part known as loop of Henle, it has further two parts. This elongated part, tubular structure, it is known as descending loop of Henle, and this one is known as ascending loop of Henle. Likewise, the last structure of the nephron, it is known as distal convoluted tubule, further followed by a broader duct known as collecting duct. So, mechanism, whenever we talk about the mechanism of urine formation, it is completed into three steps. The first one is known as ultrafiltration, the second one called selective reabsorption, and third one is known as tubular secretion. So let us first of all talk about ultrafiltration. Ultrafiltration takes place in the glomerulus of renal corpuscle or Malpighian corpuscle. So, firstly, what we see is we have artery, afferent artery, and the next we have efferent artery. The afferent artery, it is broader than the efferent artery. The blood flows through the afferent artery in high quantity, while when it needs to come out or needs to get released through the afferent artery, only a little amount of blood is released. So because large amount of blood gets deposited into the network of the capillaries or glomerular capillaries, there is the formation of large pressure called ultra pressure, which is created due to the high amount of blood present in the glomerular capillaries. So once there is increase in the hydrostatic pressure in the glomerular capillaries blood, it gets filtered through this Bowman's capsule, and from the Bowman's capsule it passes through the proximal convoluted tubule. Whenever there is creation of the hydrostatic pressure, there are also the creation of other opposing forces like the downward force created by the proteins of blood present in the glomerular capillaries. Likewise, the uh, hydrostatic pressure created by the fluid present already inside the capsule or capsular fluid. Likewise, other pressure, let's say osmotic pressure, etc. Anyhow, because the hydrostatic pressure of the glomerular filtrate or the glomerular blood is too high, which is about 75 mmHg, it gets filtered through the capsule and is finally collected in the proximal convoluted tubule. So this is all about ultrafiltration. That is, in shortcut, we can say the filtration of blood due to hydrostatic pressure from the glomerulus into the Bowman's capsule is called ultrafiltration. After the ultrafiltration, the blood the filtrate which has been passed in the proximal convoluted tubule, it still contains the nutrients, let's say, like amino acid, water, glucose, etc. Besides that, it also contains little amount of urea, uric acid, and other particles. So in this region, there is the process of reabsorption. Now, the second step of the mechanism of urine formation. Now, Before talking about this selective reabsorption, let us know what is peritubular capillary. Now here we see the efferent artery, which is collecting little amount of blood and releasing out from the glomerulus. So these tubules, these parallel tubules, and these all together, these tubules, they form a peritubular capillary because they surround the entire renal tubule. Here in the diagram, it is shown away from the renal tubule, but in fact these tubules are in a very close contact of this renal tubule. So this is called peritubular capillary. So the nutrients which were present in the proximal convoluted tubule, those nutrients, about 2/3 part, is absorbed by these peritubular capillary in the proximal convoluted tubule. Now, as the filtrate flows down in the loop of Henle, its nature and concentration changes as most of the particles which were useful has been reabsorbed by these peritubular capillaries in the area of proximal convoluted tubule. As the filtrate moves in the loop of Henle, it becomes much more hypertonic, which was previously isotonic in the proximal convoluted tubule. Now here in the descending loop of Henle, the wall of the descending loop of Henle, it is permeable to water. So as shown by the arrow, the water is released from the descending loop of Henle, but it is impermeable to the ions like sodium, chloride ions, etc. As this hypertonic fluid further passes into the ascending loop of Henle, the ascending loop of Henle is little bit permeable to the water, and from the filtrate of the ascending loop of Henle, the ions like sodium and potassium it releases, and it is being further absorbed by the interstitial cells of the kidney, or let's say by these peritubular capillaries. So in this way, still the fluid remains hypertonic in this region. As the fluid further passes into the distal convoluted tubule, the ions like sodium, potassium, and chloride are reabsorbed from the filtrate back into the peritubular capillaries. From the distal convoluted tubule, when the hypertonic fluid or filtrate still passes into the collecting duct, the ions like sodium are further being reabsorbed in this area. So this is all about the selective reabsorption. It has been in shortcut shown here in the written form. After this, the third step is tubular secretion. So where does this tubular secretion occurs? Tubular secretion takes place in the proximal convoluted tubule, in the ascending loop of Henle, and in the distal convoluted tubule. So in the proximal convoluted tubule, what happens? These peritubular capillaries inside their blood, in these capillaries, the blood still contains some useless or harmful substances like, let's say, drugs, uric acid, H plus ions, K plus ions, etc. So, talking about the proximal convoluted tubule, here in this area, the peritubular capillaries produce H plus ions and release into PCT. Likewise, in loop of Henle, in the ascending loop of Henle, the H plus ions is being released into its filtrate from the peritubular capillaries. Likewise, talking about the distal convoluted tubule, the useless materials like drugs, uric acid, K plus ions, H plus ions, they are released from the peritubular capillaries back into the filtrate of distal convoluted tubule, and which further finally moves to collecting duct. So, in this way, if we see the nature of filtrate, which was previously here, it is quite different with the nature of filtrate collected in the collecting duct. As the hypertonic filtrate comes to the collecting duct, the collecting duct too absorbs water and it becomes much more concentrated, and it is released out through the urethra finally in the form of urine. So throughout a single day there is the formation of about 180 liters of filtrate from the Bowman's capsule, while our body only releases about 1.5 liter of urine each day. We must know. Thank you. Hope you must have entertained the video.